All right, so to start, for those of you who may not be familiar with BSI products, we offer a full software suite for proteomic solutions, antibody sequencing, and glycoproteomics, as well as lab services. For this particular webinar, I'm going to be focusing on Peak Studio, which is our complete solution for bottom-up proteomics. So in this webinar, I really want this to be useful for both new and old Peaks users. So I'm going to do more of a deep dive into kind of our mostly used workflows, really explaining the algorithm, how they work, and giving you some tips and tricks along the way that maybe you hadn't thought of previously. So I'll start with an overview of what we offer in the Peaks 12.5 software as a whole. Then go into workflow structures. So that's going to involve kind of the DDA as well as the DIA analyses. And along the way, I'm going to explain the scoring that you're going to see in Peaks 12.5, and as well highlighting some new features in Peaks 12.5, specifically as they relate to the DIA workflow. And then I will do a bit of a Peaks demonstration, primarily going over some configuration options. And I'll end off with a label-free quantification, a workflow, and a deep dive into the parameter setup. And then we'll end off with a Q&A where if there was anything I wasn't able to get to in this talk or you want clarification on anything, you can ask questions then. So this is our Peak Studio 12.5. We are a vendor-neutral software, so of course that means regardless of what mass spec vendor you use, you're able to load in the raw data into Peaks and analyze it without having to convert it prior to uploading it into Peaks. Previously, we were focused more only on discovery proteomics. However, in Peaks 12 and 12.5, we've started getting into some more targeted methods as well. So we have the single software package with DDA, DIA for discovery, and then our targeted methods we offer currently are PRM and hybrid DIA, currently for thermo instrumentation. So in the PRM workflow, the way you set that up within Peaks is you would perform calibration. You would create a calibration curve and then additional samples that you want to quantify, and you can do kind of an absolute quantification set up that way. And hybrid DIA is a specific newer acquisition technique that combines discovery and targeted proteomics with a Peaks library, database search, DIA database search, a label-free quantification, and then a more specified PRM quantification of targets that you specified in the data collection. So these are our targeted methods that we offer in Peaks at the moment. And for each different data type, we have a workflow-driven analysis. So each data type is going to have workflows that are geared specifically towards analyzing that particular type of data. So I'm going to start with a bit of an overview of the DDA workflows, and I really want to highlight this because if you're not used to the newer look of Peaks, maybe you're a Peaks X Pro user, or maybe you used a previous version, we've kind of reworked the way we set up the workflows. So the way it is now is at the start of your analysis, you're going to pick your entire workflow right at the start based on the application that you want to do, whether it's a de novo sequencing, a database search, a quantification, or more application-specific glycan searches or immunopeptidomic searches, which is the deep novo peptidome. However, you still retain the flexibility of previous versions. So let's say you wanted to do a database search and then you change your mind and decide you want to add quantification afterwards. You can highlight your analysis within Peaks, select this Modify Analysis button, and then you can go back to this page, change to the quantification workflow, and then you will be able to, as long as you don't change anything about your database search, you'll be able to add on a quantification step without rerunning what was done already in the database search. So you still retain some flexibility with this workflow structure. Another tip I want to give at this point is the GPU requirements. So in our newer versions of Peaks, certain workflows can take advantage of a GPU resource. And so I'm going to explain that in a little bit more detail here. For the DDA workflows, the GPU specifically impacts the de novo algorithm, and the GPU is actually required for this algorithm to work properly. So you'll notice for our de novo sequencing options, we have our classical de novo workflow as well as a deep novo analysis too, which is more of a deep learning based method, which has FDR control on it. And in order to perform the deep novo, you need a GPU with a CUDA version greater than 12.3, NVIDIA CUDA Qt capability of greater than 8, greater or equal to 8, and a memory of greater or equal to 8 gigabytes. If you don't meet these requirements, you might notice that the deep novo workflow is grayed out. And so if you see that, that is why. The deep novo peptidome workflow does make use of this deep novo algorithm. However, if you don't have a GPU, you're still going to be able to select the deep novo peptidome workflow. It will just run the de novo algorithm in the background instead of the deep novo. But you'll still get the benefit of the deep learning based on immunopeptidomics research in the database search step. That's kind of an overview of the DDA workflows. I'm going to go a little bit more in depth into this PeaksDB workflow. This is kind of our classical workflow, and it's sort of the foundation of our DDA workflow. So I want to talk about that in a little bit more depth and kind of explain really what's going on. So Peaks uses a feature-based data analysis. Feature detection occurs usually at the first step of any workflow, and it's done at the MS1 level. So when we're talking about a feature in Peaks, we're talking about a range of M over Z values that make up an isotopic distribution for a peptide. And this is eluting over a certain retention time range. And the information in the feature includes the charge of the peptide, ion mobility information, and as well, the area will come from this feature as well, as we'll talk about in the quantification algorithm section. And I've just got this picture over here showing within Peaks how a feature will actually look. So each peptide identification, you'll also be given this graph of a feature. So you'll see outlined in the purple is the retention time range and M over Z isotopic distribution, and you'll see the MS2 isolation window here. And you'll get to hover over it and see different statistics about the feature. And you'll see a precursor profile as well. Then the other part of identification is the actual identification at the MS2 level through peptide spectrum matching. And the DDA database search, we call it a de novo assisted database search. And that's because de novo sequencing is involved in each step of the analysis. So the first step is to de novo sequence all MS2 spectra. These de novo sequencing results then inform the database search in the sense that it will help to create short lists of databases or proteins to search in the database. It's also going to be included in the scoring. So along with many other features, the similarity to the de novo sequence is going to be taken into account when scoring the quality of the peptide spectrum match. And one of these other features I want to bring up quickly while we're talking about this workflow is the deep learning boost. So this is a newer option. We introduced it in Peaks 11, the deep learning, where we will rescore peptides based on predicted retention time, fragmentation patterns, and ion mobility for a particular peptide. If your peptide spectrum match really follows kind of that prediction very well, it'll get a score boost, and then the score will be lowered if it doesn't match it very well. And so that's just another thing that goes into the scoring. For the DDA workflow, this can be turned on or off if you find it doesn't really work well for your application. And if there's no database match at the end, the de novo only results are still going to be reported. So you're not going to lose any information. You'll still get that de novo information at the end. And these de novo only results can also help our further algorithms. So the optional steps in the database search is you have the Peaks PTM algorithm, and that will allow you, in addition to the probably fewer variable PTMs you search in the database step, you can search for pretty much all the built-in modifications and peaks, which can be hundreds at a time. And the algorithm is designed in such a way that it doesn't cause a massive expansion of the search space for that purpose. And then the spider algorithm, that allows for mutations to be found in your peptides. And both of these algorithms are powered by de novo sequencing because you have good quality de novo matches or good quality de novo sequenced peptides, but maybe they don't match to a database, they only match partially. PTM and Spider can be used to see if maybe these would have a database match if we used a particular PTM or if it had a particular mutation. So that is the kind of database search workflow, how it really works in Peaks. Now I'm going to go into the scoring a little bit. So I'm going to start with the de novo results because in every Peaks result, you're going to have the de novo results reported. Initially, you will get a local confidence score at the amino acid level for each of your peptides. And this confidence is color-coded, with the most confident being in red, the least confident being in black, and colors in the middle representing different percent confidences. And hovering over a peptide in the table will get you your local confidences shown in this little chart here. And then the average of this will be taken and an average local confidence for the peptide will be reported. And you can choose a particular ALC cutoff that you want to display in the result. Next is the scoring and the database search. So there's a couple different score types you will see in your database search results that indicate the quality of the peptide spectrum match, that assignment. So the first is the -10 log P, and this is indicating the quality of this peptide to the spectrum. So the peptide spectrum match. The p-value can essentially be read as the probability that the match is false. Many attributes go into this score, such as the precursor mass error, the fragmentation that's present, the length of the peptide, similarity to the de novo sequence, etc. And then -10 log P kind of flips it just to make it a little bit more easy to read rather than a very small p-value. And then additionally, you'll also see an A score specifically for PSMs that have a variable modification on them. And that's talking about the positional confidence of the PTM. How confident are we that we got the right location for the post-translational modification? And this can also be read as a -10 log P value, where P equals the probability that the position is false. So in both cases, if you think of a -10 log P of 20, that would be a p-value of 0.01, and so a confidence of about 99%. So a score over 20 is usually considered very good. Another scoring method that Peaks uses is FDR control. And Peaks uses a specific target decoy fusion method to estimate FDR. And this decoy fusion method helps get around common pitfalls in target decoy calculation, including if there was a multi-round search or if protein information is used to rescore the peptide matches. And the way it works is the target sequences and the decoy sequences are concatenated together, and then they're searched. And of course, you find your number of decoy hits, and you take your total number of target hits, and you can use that to estimate the number of false target hits that you would expect in your peptide, because you would expect it to be approximately equal to the number of decoy hits that you get. So we offer a few different options for filtering in Peaks. So I'm just going to take a minute now to explain kind of when you should use each option, what we recommend. So if you have an experiment where maybe you have only a few proteins in the database, maybe your sample is very pure, you're not getting very much variety in terms of the proteins, or if you're using maybe a small peptide database, we would recommend peptide spectrum match, -10 log P or FDR. And this is just because using the peptide spectrum matches rather than just the unique peptides, that gives you kind of more numbers to work with for the FDR control. It's usually gets you a slightly better option. Minus 10 log P cutoff is also good for troubleshooting purposes. For example, if you're searching in peaks, you're expecting to find a particular protein and you're not able to find it, you could always take this minus 10 log P right to 0 just to remove the filtering completely, search in peaks and see, does peaks identify this at all? And maybe if it does, is there a reason it's getting such a poor score? Like, what's the wrong? Maybe it's not fragmenting enough. Maybe there's another reason going on. But I kind of like playing around with minus 10 log P for troubleshooting purposes. And then finally, for general proteomics samples, we typically recommend a peptide FDR, typically around 1%. So now I'm going to move along and talk a little bit more about the DIA workflows in Peaks, and as well the new features in Peaks 12.5. So for the DIA data type, we have 3 different workflows. The identification workflow, which I'll talk a little bit more about in a minute. The quantification workflow, where you can do label-free quantification, and deep novo peptidome, which is the immunopeptidomics focused workflow specifically for DIA. And this is a slightly newer one. We introduced it in Peaks 12. But new in Peaks 12.5, the first thing I want to talk about is the increased speed. So we've been working quite a bit to improve our DIA analysis speed. And if you look at this chart here, if you focus on the left bars here, this compares analysis time in Peaks 12 using a GPU and Peaks 12.5 also using a GPU, and then the speed in Peaks 12.5 without using a GPU, so only CPU resources, because the DIA workflow is one of the workflows that makes use of the GPU resource. Unlike the Deep Novo, the GPU is not required for DIA analysis. However, it does speed up certain steps of the analysis. And prior to Peaks 12.5, not having a GPU resource could increase your analysis time by quite a few hours, actually. So it has been quite a bit improved in Peaks 12.5. As you can see, it's only a little bit longer than it would be in Peaks 12 with a GPU. So if you were previously maybe avoiding the DIA workflow because of analysis times, maybe you didn't have that GPU resource, it might be something worth trying again because we have improved that speed. And also, if you do have a GPU, the analysis times have been further improved in 12.5 compared to 12. The next thing we added in for Peaks 12.5 DIA is a more informative LCMS view. And you can see that here you can access this graph under the data refine section. So after that step is finished in your analysis, you'll be able to click on your samples and see the LCMS map. You're able to check off to show the precursors. Precursors is really the same thing as features, just for DIA. Your MS/MS spectra, your IDs with and without precursors. So if you get an identification with no feature, you can differentiate between those two. And also de novo tags, and these are all going to be color-coded, so you'll be able to see it easier. If you click on an identification, it's going to bring up this little chart here that shows the fragment ions as a function of retention time. And if you were to hold control on your keyboard and click on a different peptide, it will bring up the 2 peptides together and you'll be able to see the overlapping fragment ions, again, that are color-coded. The final new feature we have for DIA in Peaks 12.5 that I'm going to highlight today is support for ZT scan. So this new technology by SciEx, we've been working closely with them to bring this new feature in Peaks to you. And what we can see here for ZT scan results in the feature view, we can see the Q1 dimension. So that will be displayed in Peaks, and as well, it's incorporated into the peptide scoring for the result. Okay, so now I'm going to go back to kind of the DIA identification workflow, and I want to explain how it's different than the DDA workflow, because it's handled a little bit differently due to the different data structure. So for DIA, there's 3 different steps to the workflow: the library search, the database search, and de novo sequencing. Each of these steps can either be performed in sequence or on their own. So each step is optional. You don't have to, for example, do a library search. You could just skip straight to database search if you don't have a spectral library. But generally, the way it works is if you do a library search, anything that you identify in that step is going to be reported as a library peptide. And then anything not identified is going to move on to the database search. Anything that passes the filters will be reported as database. And then anything that doesn't pass will be de novo sequenced. And that will be reported with the ones that pass your ALC threshold that you set. And they'll be reported as de novo peptides. And then the reason we have kind of this workflow setup, it's a little bit opposite of what it was in DDA. In DDA, you started with de novo sequencing of all spectra, and database search, the de novo, is last. And this has to do with the search space. So we start with the spectral library search at the beginning when the data is the most complex. There's the most unknowns to identify. The library search makes that initial search a little easier. And then the database search is a little bit more complicated to do. So that comes in second when some of the spectra have been kind of eliminated from the library search. And then finally, the de novo sequencing, which is the most difficult to do, is done when the least number of identifications need to be done. And overall, by doing all 3 of these in sequence, you're typically getting a better proteome coverage and increased accuracy. I have an example here showing a direct database search versus a library plus a database search. And you can see that with the library plus database search, you are typically getting more identifications compared to just the direct database search. So now I'm going to do a bit of a peaks demonstration. Going to hop into the software. So I'm in Peak Studio 12.5 now, and I'm going to focus on the configurations tab. So under configurations, the first thing I always like to direct users to look at is this performance configuration, because it's important that this is set correctly. So under the performance configuration, you'll see your total computer resources under here, as well as the total allocated resources for Peaks Studio. So typically the threads, Peaks is going to allocate resources for you automatically, and usually it does a pretty good job if your computer has sufficient resources. It'll allocate usually about 80% or so of the resources. You do want to be careful if you're running multiple different resource intensive software on the computer. You want to make sure you're leaving enough threads for your other tasks. But this is a pretty good setup if Peaks is really the main thing that you're running. So you have your total number of threads and then total allocated memory. It's important that you allocate the correct amount of memory. So the general rule of thumb for doing that is to take your number of allocated threads, multiply it by 2, and then add between 8 and 16 gigabytes to get the total allocated memory. Allocating less or more memory than this can sometimes lead to issues during analysis or viewing results. You could get out of memory errors. So this is usually the first place we look if a user is experiencing that. Detached service JVM helps for Peaks to multitask. We usually recommend leaving this enabled. Sometimes we'll advise users to disable it if they're running a search maybe with a really, really large search space and it's getting memory errors partway through. Sometimes we will advise that, but typically, for most cases, we recommend leaving this checked. And then you'll see your GPU resource down here. If you have this checkbox available, it means you have a compatible GPU on your computer. It's going to show for each requirement, it will show what your GPU resource has. If any of these don't meet the requirements, it's going to show up in red. And you'll notice this compute capability, it says minimum of 5, and I told you 8 before. It's because 5 is the minimum for DIA to take advantage of it. So if you only have a compute capability of 5, it's going to still allow you to use it because it will work for DIA. But for Deep Novo, it needs to be 8 or over. And so that's the performance configuration. I also want to highlight, just going to go in here, some of the other configuration options that maybe you haven't noticed before. So first, you can edit kind of your input file directories, your project directory export, so you don't have to manually set it each time. I find this is useful to set before you start. If you want to configure custom enzymes, you can also do it from here. Peaks comes with a built-in enzyme list, but let's say you have a scenario where maybe you have trypsin and lyc as and enzymes within the same sample. Peaks doesn't let you set 2 enzymes at a time. So what you can do is set an enzyme name to be 2 enzymes and then put 2 rows each with the digestion rules. So that's how, and then you would just press new, you would add the rules and then add update to add it to your enzyme list, and then it's there for you to use. Similarly with modifications, if you click, you can either click new or you can duplicate, and you can kind of copy the formatting from a different one. So you would just give your PTM a name, set the formula in this particular way that's shown here, give it the monoisotopic mass, which residues can be modified. You can set this as the letter X to do anywhere. And then peptide or protein, N term or C term. You can also define neutral losses and signature ions. So Peaks can look for these, and then it will score your PTM higher if it also finds these neutral losses and signature ions. The Peaks PTM algorithm can actually search for signature ions on its own as well. So if you don't know any signature ions, you could have Peaks look for it itself. And then there's quite a few other options you can change. You can change some algorithm settings with PTM confidence search. You can add custom quantification methods, precursor and reporter ion. You can configure custom databases. Just in the sake of time, I'm not going to go too in depth, but there's quite a few different things you can customize here. And then one last thing I want to show in Peaks is just a general result structure. So when you finish an analysis, this is what you're going to see. This was a DDA Peaks DB search. You'll see your data refine button here. You can click on a particular sample to open the refined data view. You can click here to get your de novo result without any influence from the database search, because this will pop up as soon as the de novo is done, even before the database search is finished. And then you'll get your database search too. And the way that's structured is you'll get a summary page full of statistics, a protein coverage tab, a peptide tab that shows your MS2. You can swap between the predicted spectra and the annotated spectrum. Modified peptides, that shows mirror plots of modified and unmodified. Your de novo only, so peptides that did not make it into the database search. And then just a feature view. So that's kind of the outline of the results. So at this point, I'm going to go back into the presentation for the last section where I'm going to talk about quantification. So I'm going to focus on kind of the setup for label-free quantification. I find users often have a lot of questions about this, so that's why I chose to talk about it. Many of these kind of ideas and parameter settings may also in some ways apply to the labeled quantification too, but I'm going to, just for the sake of time, focus on the label-free quantification. So this is just showing the general DDA algorithm for LFQ. It starts with the feature detection, which that actually happens at the very start of the workflow. We already talked about this. And then the feature identification association is just taking your MS2 matches and associating it with the MS1 feature. Now in DDA, you can actually choose a feature-based quantification instead of the identification-based, where if you check this, this step is going to be performed at the very end, and you're going to be able to quantify peaks that don't necessarily have a database match. But this is the default, is to have this step second. Then peaks will do retention time alignment and feature matching. So it's going to line up the samples, see which features match to each other. And then a set of features that match across different samples is called feature vectors. So that's the terminology that is often used in, the peaks user manual if you're reading about LFQ options. And then the relative abundance calculation will be done. Peaks uses area under the curve calculation, and it sums the area from each isotope. And then finally, you'll do a significance assessment, which is the ANOVA calculation in peaks, or you could also set an FDR. And then I'll also point out this match between runs option, because this is a little bit different when you compare DDA and DIA. So in DDA, this match between runs happens at the quantification level, where let's say you have two features that line up. In sample one, that feature has an MS2 identification. And in sample two, it does not have an MS2 identification. Maybe there was no MS2 was taken at that point. Maybe it scored poorly and wasn't reported. So if Peaks notices that, okay, these features line up, it can actually transfer the first identification to the second sample, and then you're going to get less missing values. That way, you're going to be able to quantify more of the same peptide. So with DDA, you set that up at the quantification level. For DIA, and this is a newer feature introduced in Peaks 12, is you can set match between runs at the identification level. So you would choose it before you pick your library search parameters. And so typically for DIA, we recommend, because it's at the identification level, we only really recommend using it if your samples, if you're expecting to have very similar protein identifications across the samples. If you have very different identifications, it sometimes doesn't give you as good results. So that's just a tip for match between runs for DIA. And I'm going to go a little bit more into the filter options in LFQ. The first step is the peptide filters. So this is saying, what do we want to include in the quantification in Peaks? Peaks 12.5 by default is usually set up to pretty much allow everything in to be quantified, and it's up to the user to put more restrictions on quality and what you want to really include. So there are some options that are a little more self-explanatory. You can filter by an average area or a charge. Can also filter by quality, which what quality means is quantifiability. So Peaks is going to look at M over Z similarities, retention times, chromatogram shapes, isotope distribution shape, signal to noise ratios, feature intensities across the feature vectors. And if these are very similar and they're very intense features, it's going to have a higher quality score because we're a little more confident in the quantifiability of this. So I just have an example here. This is what a very good quality feature vector set would look like. This one down here is more of a low quality one. You can see the shapes are irregular and not very similar, and there's not as much of an overlap. So you can set Peaks to filter out ones that look a bit like this. We typically recommend, it'll depend on your data set, but usually users will start with quality score of maybe like 5 to 10 and kind of increase it incrementally as needed. The other option I want to talk about that's very important is peptide ID count, and have at least a certain number of confidence samples per group. These mean very similar things, with the peptide ID count meaning how many times was this peptide identified in each group, not necessarily quantified, because you can have an identification without a feature. And this one down here, have at least a certain number of confidence samples per group, is talking about how many times a protein was quantified per group. So I have this little example here showing sample 1, 2, and 3, which consists of one group that all have an area of 0. It essentially was not found in those 3 samples. But then sample 4, 5, and 6, it was found quite consistently within that group. If you have at least one confidence samples per group or peptide ID count to 1 or more, these types of samples are going to be completely filtered out. So if you are expecting maybe protein identifications to exist in one group but not another, but you still want them to visually be included in the quantification, then you should set this to 0. That way you won't exclude these cases. And then use in-group coefficient of variation filter is a good option that kind of filters so you don't get too much variation between samples within a group. So it's going to actually do a calculation. It's going to set a threshold for your specific data set. And it's going to say, okay, if it's varying, beyond this threshold within a group, don't include it in the quantification. So if you're concerned really with great reproducibility between samples within a group, this is a good option to check. And then finally on peptide filters, this is a DIA specific option. The quantification preference, if you choose increased sensitivity, so if you check this box, it's going to change the error tolerance to set that tolerance a little bit higher to let a little bit more in, with the idea that you might get more quantified peptides. They might not be as accurate, but you're going to get more quantification. So you kind of get that control whether you want to have a more accurate result or a more sensitive result. And next is the protein filters. This is actually, we've quantified all the peptides. Now, how can we choose representative peptides to calculate the protein ratios? And this is handled differently for DDA and DIA. So for DDA, the top 3 peptides are used. And the top 3 peptides indicate, in the absence of any other filters, which ones have the average highest area. So those ones will automatically be used, again, in the absence of any other filtering. And it's done at the precursor level. Whereas DIA, it's actually going to use all usable peptides, but it's going to choose the top 3 fragment ions. So it's actually quantifying at the MS2 level, which is different than DDA. And then looking at these last couple protein filter options, modified form exclusion, if you happen to have a peptide that has both a modified and an unmodified form, peaks will exclude both of them from the ratio calculation, because the concern is if you use one over the other, it might not accurately reflect the total protein expression, because the modification ratio might be different than the protein expression ratio. And so if you check this option, peaks will exclude both modified and unmodified form. If there's modified form and only a modified form, it will still be included, though, in the result if it's the highest average area. And you can also choose to remove outlier. It's based on the peptide group profile. So if you have some extremities in the group profile, or some maybe like this one, for example, that go opposite of what the rest of the samples are doing, then you can, peaks will remove that as an outlier. So if any peptides have the highest average area, but they meet one of these conditions to be removed, they won't be used. And I forgot to mention as well, with DDA, you can customize the top number of peptides that you use. And for both DDA and DIA, you can specify a minimum number of peptides that have to be there for the protein to be quantified. If you have less than the specified number, the protein won't be included in the quantification. And then the last thing I want to touch on before we wrap it up is normalization methods, because this is important that you select the correct normalization method when doing LFQ. So TIC normalization is really only if you have consistent protein expression across all of the samples. So if you have the majority of maybe housekeeping proteins unchanging across all of the samples, and maybe approximately the same number of up and down regulated proteins, across the different samples. Essentially, you're looking for this total ion current to be the same across samples. That's what you're expecting. And TIC normalization is good. It'll calculate the normalization factors, assuming that these should match. Manual normalization is going to use the same logic as TIC, except you get to set the expected ratios. So if sample 1 is double the concentration of the next sample, then you can set that the expected ratio, and it will calculate with that assumption. Internal standards is another option that it requires some certain conditions for it to work. You have to have previously run a quantification to get a list of proteins that are available to be used as internal standards. You have to modify that analysis, and then internal standard proteins will become available. You'll be able to search for specific proteins, check off the ones you use. You can use as many of the proteins that you want, but you're assuming that these proteins are unchanging across the samples, or you can set as well expected ratios. So you can say in sample 1, it should be this, in sample 2, it should be twice that, for example. And then the final option is no normalization. And this is for conditions that don't really meet the above. If you don't have consistent protein expression, if you're not expecting the TICs to really be the same, if you're expecting very different proteins from sample to sample, then no normalization is a good option. And so that's it for label-free quantification. And so with that, I'm going to wrap up the talk as we're almost at time. Before I open up the question and answer section, I'll just point out a few ways that you can get in touch with us. So our technical support, easiest way to get in touch is to email support@bioinfor.com. We're happy to help with troubleshooting, analysis, setup, just general questions about peaks, how things work, how to interpret results. We're happy to help as well. We always welcome feedback and feature suggestions and requests if there's something really specific you're hoping to see in peaks in the future. As well, I have the emails and phone numbers for the software sales and pricing. If you're interested maybe in doing a Peaks 12.5 trial, you can reach out to sales, and as well our lab service contact if you're interested in some lab services. So with that, I'm going to thank everybody for listening to my talk today, and I'm happy at this point to take any questions.